So today's lecture is going to be pretty short. Yesterday we finished up talking about peptide synthesis, and we said there's a bunch of problems that had to be overcome to link amino acids together. The first one was that you can't just mix two amino acids and have them linked together because they'll just do proton transfer. So we had to turn that OH into a good leaving group, and we talked about these special peptide coupling reagents, one of which is DCC, and we went through the mechanism looking at that. We said the nice thing with this is you can purify your new dipeptide using just simple acid-base extraction or an aqueous workup. It's pretty simple, and it has a high efficiency, so pretty high yields with this coupling. For the next problem, we said you can't just mix two amino acids together with a peptide coupling reagent, or else you get a mixture of 4 different products. And the way to prevent that from happening is to protect your amine group on one of your amino acids and your carboxylic acid on the other. And we said the Boc group is normally used for amines, and we took a look at Boc anhydride for installing that, and TFA for removing it. And then we said for protecting groups, the easiest thing to do is convert your carboxylic acid to an ester. Esters don't do proton transfer chemistry. A variety of esters can be used, so you might see different ones on sapling. However, benzyl esters are really easy to remove using a variety of conditions. And then problem 3 was where we finished, and we talked about Fusion. It's the HIV drug made of 36 individual amino acids linked together, purely synthetic. I think the original synthesis took about 114 steps, and that means that yield is a huge problem here. So that's the last bullet point I wanted to talk about. It is really, really, really challenging to make these in high yields. Does anybody know the solution? I think we brought it up briefly during the last lecture. Make a cell do it? Yeah, actually that is one thing. So cells are really good at making proteins. So proteins are polypeptides that have been kind of folded up into a special shape or structure. Chemists oftentimes want to make artificial polypeptides that don't have a discrete shape, so cells aren't very good at doing that yet. The easiest solution at this point is automation. So automate this process. And a huge amount of money has been poured into this, developing automated techniques for making polypeptides to the point where you can basically type in a sequence of polypeptides, email that to a company, and they'll ship you that custom polypeptide in a matter of 3 days. It's really efficient nowadays. And I'll show you the general scheme for how this works. However, every automated system is slightly different from one another, so I'll just show you one of the most generic ones. And this is called the Merrifield synthesis. He was the chemist that helped develop this system, but it's really straightforward. And what he does is, or what he did was he had a polystyrene resin. And so polystyrene is a big, long polymer. And on this polymer down here, he's got a bunch of these alkyl groups, right? So he's functionalized his polystyrene with these leaving groups. If you treat this with a Boc-protected amino acid, you can imagine really quickly this oxygen is going to do SN2 chemistry and kick off our leaving group. You don't have to worry about writing down this full mechanism. I just want you to more know the idea. And then what do you think you do next if you want to remove this Boc group? TFA, exactly. So the next step of this automated sequence is treat this with TFA. When you do that, you now have something that looks like this. I'm not going to, oops. Actually, I'll leave this as NH3+. I'm not going to draw the full polystyrene just for the sake of time, but you can pop off that Boc group, no problem. In the next step, in the automated sequence, you take your new Boc-protected amino acid, and you typically have some sort of peptide coupling reagent, so maybe DCC, something like that. And because this has a free carboxylic acid end, and you've got an amine over here, you can quickly link those together into a dipeptide. Once you have your dipeptide, you want to remove the Bach end of this amino acid. So again, you'll do TFA. And then you can repeat this over and over again. And theoretically, you can use this method to make polypeptides that are hundreds of amino acids long to get these super exotic pharmaceutical drugs. And this is still how the pharmaceutical industry does a large portion of this. However, more and more they're facilitating cells to make short polypeptides, taking those and then linking them together. So they do kind of a hybrid between synthesis and biochemistry. At the very end, though, the tricky part is removing this polypeptide strand from your plastic, that polystyrene. And in order to do this, you treat it with hydrofluoric acid. That'll cleave your peptide from the polystyrene resin, and you get a free polypeptide. This is hugely efficient, like higher than 99% yield for every step. So that really cuts down on the waste. It cuts down on the cost of hiring a human to do every step. The purification is really easy and straightforward. It's highly efficient. In fact, this similar methodology, people looked at this and they said, well, why can't we do something like this for just any synthesis? And what's happening more and more is chemists are being displaced by robots. We're essentially being outsourced. There are new programs and automated techniques for making custom small molecules as well. The really cool thing with this that people are getting fascinated about is they're doing AI systems as well. So what they're doing is they're coupling AI computers to every journal article on the planet. And the AI computer will scan every one of those articles, look at each synthetic route, and quite often they'll say, we won't count this as a real synthetic route unless it's been done at least 3 times with a yield above 80%, something like that. And then figure out the best way to make something, plug it into a computer, and a computer will actually generate the best synthetic route and then plan the synthesis, and you can automate it. The future of organic chemists is a little bit strange in that regard. Some people are arguing that organic chemists will be entirely displaced by automation. Other people are saying that's like saying engineers will be replaced by robots. It's not entirely the full story. The truth is somewhere in the middle. So that's kind of where I wanted to end today, was more of this advanced technique for making unique custom polypeptides. If you go on and work in a biochemistry lab, you'll see these automated techniques over and over again. You can do the same technique with RNA and DNA and just custom order whatever sequence you want. It's pretty powerful stuff. All right, so the last bit of organic chemistry is just our chapter review. Like I said, the synthesis of amino acids, these first 3 aren't very commonly used. The first one was the Hell-Volhard-Zielinski modification, where you do alpha bromination of a carboxylic acid and swap it out for an amine. Why aren't these used very often, these top 3? Anybody remember? Yeah, if you think about it, we've got a stereocenter here, here, and here. If you have to purify that, you're immediately going to lose 50% of what you made. So the top 3 ones here are more old school syntheses that aren't commonly used. The enantioselective synthesis is generally what's used today to make exotic custom amino acids, meaning ones that aren't naturally found, ones that you can't just order off the internet. And you do have to use a chiral catalyst to get these enantiopure. The other thing we saw was the coupling of individual amino acids using peptide coupling reagents, primarily DCC. And then we saw how to install protecting groups for carboxylic acids and for amines. So really the big ones I want you to know are the protecting groups. Those are essential for biochemistry, knowing what they're used for and how to install them and remove them. And I want you to know the general idea behind peptide coupling reagents. I'm not super picky about the mechanism because, to be perfectly honest, there's about 24 of these on the market. DCC isn't the most commonly used peptide coupling reagent. And that's it, believe it or not. The remaining time that we have here, I want you guys to work in small groups and to try to go through the practice exam. What I would encourage you to do is not to open your notes, but use each other. Trying to teach each other how to solve problems is incredibly effective. So try to do it as much as possible without notes. And then tomorrow we'll go over the key.